And welcome to a video going over cancer. So I believe some of you, if not all of you, on Friday, I know as a short class, were able to play the game where I asked you to memorize these nucleotides: ATCGA, CGAAAA, CGC, ACTTA, and then write them down on your piece of paper. And then I asked you to grade your accuracy. The reason I asked you to grade your accuracy after memorizing it is cancer is nothing more than making mistakes when we copy down information during S phase in mitosis. So, cancer. Every time a cell divides during S phase, we have to duplicate about 3.2 billion nucleotides. This takes 1 hour on average, and it's a lot of Bibles or a lot of textbooks to give you a context. So, there's a lot of information being copied down. DNA polymerase, which is the enzyme that we saw um, when we talked about the proteins that make mitosis occur, is an enzyme that copies DNA from the original strand. It makes about 1 error per 10 million bases. I don't know why that comma is there, so ignore that. Or it has about a 99.9999% accuracy rating. So if I gave you a $100 bill, it's a thousandth of a penny in terms of its mistakes, which is pretty remarkable. Before the cell exits G2, the cell uses additional enzymes and proteins to proofread and fix DNA. However, still, despite using what we would effectively call really good Grammarly, I know many of you might use that on your papers, occasional errors can persist, be missed, and make its way into the next cell, which can cause issues. Cancer is just the development of accumulated mutations in the DNA of a cell. And a lot of that mutation directly impacts DNA or codes or genes that have the ability to recognize and regulate growth, as well as check its genetic code. So, again, the mutations that happen in DNA are often random, but they occur in specific genes or locations that might help cells determine what checkpoint they're in. Should they go past the checkpoint? Is there RGR, repair, growth, and regulate, not regulate, repair, grow, and replace? Is that needed? And also, as we'll talk about, ways to check genetic code. When cells become cancerous, they don't really have that ability to check growth and check their own code. So it's sort of like your consciousness going away or going awry. Your consciousness is that stream of thought, those thoughts, those ideas in your head that help you make decisions. When you lose that, sometimes you can make bad choices. So I'm going to ask that you watch this video. I'm not going to play it right now because actually, let's play it.
In this health sketch, we're going to talk about cancer, a very common condition that affects millions worldwide. Cancer can affect anyone, but risk generally increases with age. Most of us have heard of it, but do you actually know what it is? In fact, cancer is a group of over 100 diseases caused by uncontrolled cell growth. Your body is made up of trillions of cells of different types. These cells are constantly working, dying, and being replaced through a process called cell division, where the cells make copies of themselves. This process usually happens in a very controlled way, so that we maintain the right numbers of each type of cell. This is because of the DNA, a code of instructions which carefully controls what each cell does, telling them when to die or when to divide. However, errors can occur in the DNA instructions called mutations. DNA mutations can occur randomly when the cells don't copy themselves correctly, and these random errors build up over time, which is why cancer risk increases with age. DNA mutations are also caused by environmental factors that damage DNA, such as tobacco smoke, alcohol, obesity, pollution, certain infections, and sun exposure. More rarely, mutations are inherited or run in families. Mutations are often harmless, but sometimes cause a cell to copy and grow uncontrollably, which is how a cancer starts. These abnormal cells keep creating copies of themselves, growing into a clump or mass of cells called a tumor. Some tumors stay in one place and do not spread, known as benign or non-cancerous tumors, and these are less likely to cause problems. Tumours can also be malignant or cancerous, which means they can spread to other parts of the body. Malignant tumours are more likely to cause problems as they invade other body structures, affect the way the body works, or use up the body's nutrients. Tumours that have spread away from the primary site of the cancer are called metastases, or METs. Therefore, it is really important to detect cancer at an early stage, as it is easier to treat before it has had a chance to spread. Routine screening tests are used to pick up some common cancers. It's also important to see a doctor if you experience unexplained symptoms that could be a warning sign of cancer. Various investigations are used to diagnose cancer. These might include blood tests, imaging tests, and a biopsy, where cells are taken for examination under a microscope. If a tumor is found, these investigations help determine the type, size, and spread of cancer, which is called its stage. This is used to work out the most appropriate treatment. The aim of cancer treatment is to remove or destroy the tumor cells in the body while minimizing damage to normal healthy body cells. The main types of cancer treatment include surgery, where the tumor is removed from the body, chemotherapy, where anti-cancer medicine is taken to kill off cancer cells, radiotherapy, where radiation is targeted to destroy cancer cells. Other forms of cancer therapy include immunotherapy, hormone therapy, and gene therapy. Some cancer treatments also have an effect on normal body cells, particularly fast-growing cells like the skin, hair, gut, and bone marrow. This can lead to a range of side effects such as rashes, hair loss, nausea, and tiredness, though these are generally temporary and usually resolve after treatment finishes. Cancer treatment might significantly reduce the tumor size and ease symptoms, known as remission. But it is often difficult to know whether all the cancer cells have been removed or destroyed, and if some remain in the body, the cancer might return months or years later, which is called a recurrence. The overall prognosis varies a lot from person to person, depending on the site, type, and stage of cancer, and the treatments available. Due to the remarkable progress of medical science, advances in cancer therapy are happening all the time, and these continue to lead to improvements in cancer survival worldwide. We hope this health sketch has been helpful to you and those around you.
All right, so let's move on. Cancer is, again, when mutations occur within the DNA, specifically in genes that help regulate the cell cycle or limit growth. Now, a gene is a sequence of nucleotides, A's, T's, G's, and C's, that when read, their order means something to the cell. Sort of like when we put letters into words and order those words into a particular way, we create a sentence with a very specific meaning. DNA in a very specific order will create a very specific protein. When that DNA is changed, the protein will change. So, some examples of DNA mutations. The dog was happy and glad to chew a bone. This is a very normal, expected image with a very normal sentence in the context of our culture and our world. Sometimes DNA, when it's being copied by DNA polymerase, will actually have the DNA make a mistake. The RNA polymerase will put in a T instead of a G. It doesn't catch it. For some reason, the base pair proofing doesn't occur. So the dog was happy and glad to chew a phone. That doesn't look very good. The dog is probably going to get very sick. It's very sharp, so there might be some injuries. Other times when DNA is copied, things are duplicated. A section of DNA is copied more than one time. Again, this happens very rarely, but it can occur. So instead of chewing one bone, the dog is going to be chewing a bone that is made of 4 different bones. A bone, 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 bone. The DNA was duplicated. And other times, DNA can actually be inverted when it's copied. The RNA polymerase switches 2 segments of DNA around, and we get inversion. The bone is chewing the dog. I'm not really sure what's up with this teeth, but the dog is certainly being surprised that the bone is chewing it and not vice versa. So let's talk about the Rh gene. In many cells, when a growth hormone binds to a growth hormone receptor, a cell response results in the cell dividing. So a lot of you right now in your pre-adolescent, puberty, teenage years have growth hormones circulating through your body 24/7. They can be released after exercise. They can be released while you're sleeping. They are certainly age dependent. They are diet dependent. They are health dependent. But growth hormones generally bind to a cell and cause it to divide. And growth hormones are sort of in the context of a broad-based hormone. They're going to cause a lot of different parts of our body to divide. Upon reception, with between the growth hormone and the receptor protein, that ligand, that connection causes a protein called Ra to become activated using a cousin of ATP called GTP, which starts the transduction of the cell response. You remember the game of tag. 1, 2, 3, you're it, now you're it, now you're it. That overall transduction pathway will cause proteins to go into the DNA to make proteins that help stimulate the cell cycle. So growth hormones are released during your sleep cycle so that your muscles can grow and divide while you're sleeping rather than when they're being used. In mutated cells, however, a different version of Ra is created because of DNA errors. Because Ra is a protein, it has a gene that makes it in all cells. In the mutated version, Ra is always on. It is always activated. It will be turned on. It will be working without growth hormone being present. What this means is the cell will be dividing all the time, making more of the proteins that cause cell division. Some of these proteins are the cyclins that we talked about in class on Friday. Usually what happens is that Ra activates RAF, which activates ERK, and there's again that game of negative feedback. Ra turns on RAF, RAF turns on Ra, turns off Ra, RAF turns on ERK, and then ERK causes the transcription factor. This is a negative feedback loop because it's the on-off cycle. In cancer cells, however, as I was saying, Ra is always on, so it's always tagging RAF, which RAF is always tagging ERK, so we have a proliferation of cell division. Cell division is always happening. So that's again a mutation that's happening when cells are just constantly dividing because they're being tricked into thinking that there's actually a reason to grow. Another really common mutation in cells is called the p53 gene. Some proteins are activated during mitosis, and they are responsible for making sure that any errors or mistakes in the DNA are corrected. This is often happening during G2, before the G2 checkpoint, and this makes sure that any issues with our DNA polymerase are fixed. Mutations can happen because of our environment, because of our age, because of just random mistakes with DNA polymerase. So generally, for example, if you get a lot of UV light, UV index of 8 or 9, and it causes damage, your cell will detect that damage, activate p53, and p53 will cause it to be fixed. If, however, those fixes aren't possible, then the protein might inhibit the cell cycle. We know that there's these mistakes. Maybe there's damage to our DNA. We can't fix it right now, so we're going to stop mitosis. This generally happens during the G2 checkpoint. That means no cell division occurs. We call p53 the guardian of the genome, given that it stops any division from occurring when there are mistakes. It sort of comes in, it swoops in and says, hey, you can't divide. Hey, you shouldn't divide. We're going to stop it because it's not the right time. We have mistakes. We have to fix them. In mutated cells, however, the gene that codes for p53 is damaged. And so the actual DNA that codes for p53 is broken, which means that p53 is defective. It's missing parts of its ability to bind and recognize damage, so it is offline. It can't fix anything. It's like Grammarly without the internet. The program is there, but without the internet, it can't think and do the work for you to find those errors, which means other mutations are not caught, not fixed, and the cell starts dividing with all of those mutations. If P53 is working, it can do the following things in order, and this order is really important. One is it can repair DNA damage. If the damage is easy, fixable, easy peasy lemon squeezy. If you bring your car into the dealership or the mechanic and they're like, we have to replace your disc brakes because they're worn out, easy peasy lemon squeezy. We can fix that. If there is a problem with the engine, well, we're going to have to stop driving that car for a little bit. If there's bigger issues, the cell will stop mitosis altogether because there's just too many problems to fix and we have to take it off the cell offline for a little bit. Let's make sure we can fix these errors. If the car is totaled, if you get in a car wreck with a moose, if your car is at 250,000 miles and everything is broken on it, then apoptosis might be necessary. And apoptosis is programmed cell death, where the cell splits up and pops. And this is a very normal activity. We do it often when cells are getting old and dying. But again, this is useful for us to prevent cells from dividing and being broken. So a quick reminder, shout out to Kami Koba, who is on the slideshow on prom night. This is right now. What time is it? 2:45. Kami, I hope you're getting ready to boogie. So a reminder, when we get to our normal control, our CDKs, really the Rod gene is going to be really responsible during G1 to know if the cell should divide. Well, P53 is going to be making sure that we've done everything correctly. I know this video has been long. Thank you for your patience, and I hope you guys have a great weekend. See you soon.